So good morning everybody, my name is Miccio and I'm a group leader here at the Institut, and in particular the name of my lab is Chromatine and gene regulation during development. So despite this very complicated name, I would like to explain you that one of our main goals is to develop safe and efficacious therapeutic strategies for a disorder affecting the blood cells, and in particular for sickle cell disease. And as you might know, sickle cell disease is one of the most common genetic disorders worldwide. So there are more than 300,000 patients that are born each year everywhere in the world. And this disease is caused by a mutation that affects the production of the hemoglobin, which is the protein that is present in our red blood cells and whose function is to carry oxygen from the lungs to the tissues. And so in the presence of this altered hemoglobin, these red blood cells adopt a sickle shape, so they are basically deformed and they tend to adhere to the blood vessels. And so this leads to occlusion of the different blood vessels in the body, and therefore this is really a multi-organ disease. The World Sickle Cell Disease Awareness Day is very important because we need to raise awareness in the general public and we need basically to explain what sickle cell disease means and which are the challenges that every day the patient, the families and the caregivers have to face. And in particular, this is also important to promote the screening for sickle cell disease because, as I told you before, this is a disease caused by a genetic mutation, but basically the patient needs to inherit two genes, one from the mother and one from the father, and both of them needs to be mutated, and so basically the father and the mother are carriers of this gene. We say that they carry the sickle cell disease trait, which means that these carriers should be identified, and there are very simple tests in the lab that we can do to identify the carriers. And it's very important to know that you are a carrier of sickle cell disease, because I think that people need to be aware that there is a possibility to have a sick child with sickle cell disease, and they need to know which is the frequency with which basically they can have a sick child. So basically my team is working on the developing of gene therapy approaches, in particular for sickle cell disease, which means that we aim at collecting the cells from the patient and to genetically correct their defect in vitro in the lab, and then to reinfuse these cells into the patient that will then hopefully produce a normal hemoglobin. And therefore we call this approach gene therapy. And so currently we have several clinical trials that are ongoing worldwide, focused on the gene therapy for sickle cell disease, and these trials have shown very early promising clinical results. And but they also show us which are the next challenges of this kind of therapies. So basically what we could ameliorate, and in particular how we can, which are the issues that we have to face to improve the efficacy and the safety of this therapy, as well as to reduce the price. So one of our goals is also to reduce the cost of this type of therapies, because to give you an example, the current therapies cost around 2 million per patient, which is definitely something that we cannot afford for treating all the different thousands of patients that exist worldwide. And so basically the idea is also to simplify the procedure and the tools that we use to correct the genetic mutation. And of course we are working on many different approaches, and in particular Imagine and our lab is coordinating an international consortium that includes scientists from many countries in Europe, such as Italy, France, Netherlands, Germany, even Israel, and also, for example, the EuroBloodNet, which is a European infrastructure that includes patient association. And so this international consortium aim at comparing the efficacy and safety of all these different tools that we can use. And of course, there are now many different tools, genome editing tools that we can compare, but we need to be aware of their efficacy, but also of their safety. And so the goal of this consortium is really trying to define some assays that can allow us to define and identify the best treatment for the patient in terms of efficacy and safety. In particular, this current procedure that includes transplantation, the transplantation is very cumbersome and is not definitely something that you can do in countries such as in third world countries where there are not so many transplantation units. And so the idea is now to do an in vivo therapy in which we develop a tool which could be a vector, let's say, that could directly go to the cells of the patient and genetically correct them. And although if this is a very difficult task because the cells of the patient are very hard to be reached because they are in the bone of the patient, this is still something that we would like to pursue to be able to, in the next year, to reduce the cost of this therapy.